Alright, this is for Mrs. Zimmerman's class, chapter 5, cell division. The first segment, is cell increase and decrease. Cell division increases the number of somatic or body cells. Cell division consists of mitosis and cytokinesis. Apoptosis, or programmed cell death, decreases the number of cells. The cell cycle is an orderly set of stages that takes place between the time a cell divides and the time the resulting cells also divides. The stages of interphase. Most of the cell cycle is spent in interphase. This is when the cell carries on its usual functions. Interphase is divided into 3 stages. The G1 stage occurs before DNA synthesis, and the S stage includes DNA synthesis, and the G2 stage occurs after DNA synthesis. The mitotic stage following interphase, the cell enters the mitotic stage when complete 2 daughter cells are present. Apoptosis. During apoptosis, the cell progresses through a typical series of events that brings about its destruction. In chapter 5, section 2, control of the cell cycle and cancer. Eukaryotic cells have evolved a complex system of both internal and external signals for regulation of the cell cycle. Carcinogenesis, the development of cancer, is a multi-stage process involving disruption of normal cell division and behavior. Proto-oncogenes encode proteins that promote the cell cycle and prevent apoptosis. Tumor suppressor genes encode proteins that stop the cell cycle and promote apoptosis. Just section 3. Maintaining the chromosome number. At the time of cell division, the chromatin condenses to form highly compacted structures called chromosomes. Each species has a characteristic chromosome number called the diploid, or 2n number, because it contains 2 a pair of each type of chromosome. The overview of mitosis. Mitosis is nuclear division in which the chromosome number stays constant. A 2n nucleus divides to produce 2 daughter nuclei that are also 2n. Before nuclear division takes place, DNA replication occurs. A duplicated chromosome is composed of 2 sister chromatids held together at a region called the centromere. Mitosis in detail. During mitosis, a spindle brings about an orderly distribution of chromosomes to the daughter cell nuclei. Mitosis in animal cells. Mitosis is arbitrarily divided into 4 phases. Prophase, the chromosomes are now visible. Spindle fibers appear and the nuclear envelope begins to disappear. Metaphase, the chromosomes are attached to spindle fibers and line up at the metaphase plate. Anaphase, the sister chromatids separate, becoming 2 daughter chromosomes that move toward the opposite poles of the spindle. Telophase, the spindle disappears, the nuclear envelope reforms, and the chromosomes decondense. Mitosis in plant cells. Exactly the same phases occur in plant cells as in animal cells. Although plant cells have a centrosome and spindle, there are no centrioles or asters during cell division. Cytokinesis in animal and plant cells. Cytokinesis usually accompanies mitosis, but is a separate process. Cytokinesis in animal cells. A cleavage furrow and indentation of the membrane between the 2 daughter nuclei forms during anaphase. A contractile ring pinches the cleavage furrow, separating the 2 cells. Cytokinesis in plant cells. The rigid cell wall does not permit cytokinesis by furrowing. Instead, cytokinesis in plant cells involves building a new cell wall between the daughter cells, reducing the chromosome number. Meiosis reduces the chromosome number in such a way that the daughter nuclei receives only one of each kind of chromosome. The overview of meiosis. When a cell is diploid, the chromosomes occurs in pairs. The members of a pair are called homologous chromosomes. Meiosis is divided into meiosis 1 and meiosis 2. Meiosis 1. During meiosis 1, the homologous chromosomes separate. Meiosis 2 and fertilization. During meiosis 2, the sister chromatids separate. The daughter cells mature. I need about 2 minutes. The daughter cells mature in gametes or sex cells, sperm and egg that fuse during fertilization. Fertilization restores the diploid number of chromosomes. Meiosis in detail. Meiosis requires 2 nuclear divisions and results in 4 daughter nuclei, each with half the number of chromosomes as the parental cell. During meiosis 1, genetic recombination of the parental genes occurs through 2 key events: crossing over and independent assortment. The first division, meiosis 1, has 4 phases called prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1. In prophase 1, synapsis occurs, followed by crossing over. Metaphase 1 and anaphase 1. During metaphase 1, the homologies align at the metaphase plate. Independent assortment occurs when these pairs separate from each other. Telophase 1. In some species, the nuclear envelopes reform and nuclei reappear. Interkinesis. The period of time between meiosis 1 and meiosis 2 is called interkinesis. No replication of DNA occurs. Second division. During the second division of meiosis, sister chromatids separate from each other. 4 daughter cells result. The importance of meiosis. Meiosis produces haploid cells that are no longer identical to the diploid parent cell. Crossing over and independent assortment result in different combinations of chromosomes than the parental cell. Upon fertilization, the combining of chromosomes from genetically different gametes helps ensure that offsprings are not identical to their parents. Comparison of meiosis with mitosis. Occurrence. Meiosis occurs only at certain times in the life cycle of sexually reproducing organisms. Mitosis occurs almost continuously in all tissues during growth and repair processes. Tables 5.1 and 5.2 compare meiosis 1 and mitosis 2 to mitosis. Comparison of meiosis 1 to mitosis. During meiosis 1, but not mitosis, homologous chromosomes pair, undergo crossing over, and separate. Comparison of mitosis 2 to meiosis. The events of meiosis 2 are just like mitosis, except that the nuclei contain the haploid number of chromosomes. Chapter 5, Section 6, the human life cycle. The human life cycle requires both meiosis and mitosis, spermatogenesis and oogenesis in humans. Spermatogenesis occurs in the testes of males and produces sperms. Oogenesis occurs in the ovaries of female and produces eggs. Spermatogenesis, primary spermatocytes, which are diploids, divide via meiosis to produce 4 spermatids, which mature into sperm. Oogenesis, primary oocytes, which are diploids, divide into meiosis 1 to produce 1 secondary oocyte, which receives almost all the cytoplasm and apolar body, a non-functioning cell. If the secondary oocyte is fertilized by sperm, it completes the second meiotic division, in which it again divides unequally, forming the egg and a second polar body. If the secondary oocyte is not fertilized, it disintegrates.